Hi, everyone, and thanks for tuning in to this very special episode of the Fandom Science Podcast. Today's episode, as you guessed from the title, is on gene doping, which has been kind of a hot topic in recent years as we start to talk more about the limits of human performance and what sports could look like in years to come. And there's probably no one more suited to talk about this than my guest today, Dr. Matthew Porteus, who is not only a practicing medical doctor and a researcher at Stanford University, but he's also a part of the gene and cell doping expert group that's been employed by the World Anti-Doping Agency specifically to help them fight off gene doping and catch athletes who might be using it. So today we talked about what gene doping is, how it works, what kind of genes might be desirable for athletes, do we currently have the technology to do gene doping, and what kind of challenges come in the way of athletes who are considering using this, and what dangers could gene doping have on an athlete's health. As always, I really hope you enjoy this episode and take something useful from it. And if you do, don't forget to share this with someone else who might also enjoy it. And don't forget to subscribe to the podcast for the best sports science content. Enjoy. So before we get into the gene doping talk and the hardcore, nitty-gritty stuff, I would just like to start by asking you about your work as a medical doctor, and specifically you work in gene therapy. So what got you interested in that specific area as opposed to, the various other forms of treatment?
Yeah, so I am a doctor. I'm a pediatric hematologist, so I take care of kids who have blood diseases, and I also take care of kids undergoing bone marrow transplants. But when I was in medical school, I started taking care of patients who had genetic diseases of the blood, like sickle cell disease, and it really inspired me to think about, well, we knew so much about the genetics of these diseases, but we offered no therapies at the time that was based on that understanding. Mm-hmm. And so I've begun a career-long journey of trying to develop what we now know as genome editing as a way of repairing genes that cause disease. And it's been exciting to see the slow and steady progress that then was really accelerated a few years ago by the discovery of the CRISPR-Cas9 system, which has really enabled us to make progress to a point where now we see for serious medical diseases, the use of genome editing entering the clinic, and there are going to be even more in the next few years.
So what's the CRISPR-9 system? Can you explain a little bit what that means?
Yeah, so CRISPR-Cas9 is a description of a system that was discovered in bacteria. So just as we have immune systems to protect us from infections from viruses and bacteria, and our immune system is designed to adapt to the various different viruses we might be infected by. As we all know right now, the COVID situation is caused by the problem is we had no pre-existing immunity, and so we have to rely on our adaptive immune system to develop immunity before the virus makes us ill. It was surprising and really quite amazing to learn that bacteria have a similar adaptive immune system. And basically what the bacteria do is when they get invaded by a foreign, phage, which is the virus for bacteria, they basically capture some of the phage DNA and then integrate it into its own into the bacterial genome, and then use those sequences as a way of resisting the next infection. And that structure is called clustered regularly interspersed palindromic repeats, which is the acronym CRISPR. Mm-hmm. Now, one of the key proteins in that bacterial defense mechanism is called Cas9, or CRISPR-associated protein number 9. And that's the key protein. And so the way the system works is it takes the little fragment of the DNA, turns it into a small RNA molecule, and then complexes the RNA molecule to the Cas9 protein. And the next time the RNA molecule sees a DNA that's identical to the RNA, it activates the Cas9 to cut the DNA. So what the amazing discovery was made is that you can take the guide RNA and the Cas9 protein out of a bacteria and put it into a human cell. And if you program the guide RNA with the right sequence, it can find an identical sequence in the genome and make a cut in the genome by the Cas9 protein. So we've taken a bacterial system, and it is so robust, it performs essentially the same, has the same biochemical activity in a mammalian cell. Now, the reason that's so important is that when you make a break in the genome of a mammalian cell, a human cell, a break is a very dangerous event because you're separating the chromosome. And so cells have ways of repairing that break, a lot of different ways of repairing the break. But in the process of repairing the break, we can modify the genome at the site of the break, either by creating new mutations at the site of the break or by templating the repair of the break so we create specific changes where we could fix a disease-causing mutation. So that was I hope a good overview of the system. But if not, I'm sure you can follow up with some questions.
No, that makes a lot of sense. So that's essentially how genome editing works, or gene therapy, is that the guided RNA.
Well, so let's spend a little time there. So gene therapy is an umbrella term that describes us introducing new genes into a cell to change how the cell behaves. So, for example, if a cell was missing, had a broken gene, and you delivered a new gene, it could compensate for the broken gene or the missing gene. Gene editing, I consider to be a subform of gene therapy, because gene therapy can be done lots of different ways. It doesn't have to be done just by fixing a mutation or adding in a specific change into the genome. So genome editing is a precise, specialized form of gene therapy in general.
Right. And so given your expertise in gene therapy and gene editing and this topic, so WADA, the World Anti-Doping Agency, brought you in as part of a group called the Gene and Cell Doping Expert Group. Can you describe to us a little bit what that role entails and what you help WADA with?
Yeah, so actually, just even stepping back, because of my expertise in gene editing, I've also been involved in conversations about the ethics and of whether we should do embryo editing as a way of altering that. And I think my input in that got the attention of WADA and this expert group. And so they invited me to participate because I had this broad expertise in gene editing and have been thinking about how it might be applied outside just using to treat patients. So, the group is, I think, a group that was convened many years ago. I'm just a newest member, because they recognize that one way that gene doping might occur, or one way that doping might occur, is through gene doping. And they wanted to convene people who had expertise to think about, well, how might that be done? And how might you design ways of detecting it if it was done? And so they wanted to convene a group of experts to help them think through both of those issues.
And so let me just jump back a little bit. So you're talking about your input on the embryonic gene editing, and so that's what caught their attention. So what was your input on that?
Well, my input, and the conversation is still, I'm just one person in a much broader conversation, just like the gene doping conversation, is that currently the technology is not good enough to allow us to safely and effectively do embryo editing. It's maybe good. It's good enough to start testing in what we call somatic cells, so cells that would not be passed along to future generations. But it's clearly, and actually more and more data is coming out, that it's just not good enough for embryo editing, to think about using this. Now, that being said, I think, and some people have said, okay, it's not good enough, so it shouldn't be done. Technically, it's not good enough. I think where people are thinking about though is as well, it's not technically good enough in 2020, but what happens when it does become, or if it becomes technically good enough? Is this something that we should do? And lots of people have weighed in, and I think there's some variations and about no, even if it became technically good enough, this crosses a boundary where we should never do it. I am on the side that there are a very small number of couples who might be at risk of having a child with a serious genetic disease. And in those very, very rare circumstances, you might consider doing this. Mm-hmm. But I would not see this as something that would be generally applicable or something that we should be using.
Right. So it's mostly just as like a preventative measure for possible diseases, or right?
Exactly.
Right. And so, what about WADA? Is that similar to WADA? For example, what I mean by my question is, has gene doping been done already, and the technology is good enough, or did they bring you in because in the future it's inevitable for it to happen, and so they want to get ahead of the game?
Well, as far as I understand, is we have no documented instance where gene doping has been used. Of course, what's the saying? Lack of knowledge of an unknown doesn't mean that it hasn't occurred. What we know is that it has not been detected yet. I actually think that gene editing is probably not going to be the first way that gene doping is tried. There are other ways of delivering genes that are more advanced that people might try. I think that my personal opinion is that I suspect it has not. I suspect it has been tried, but not successfully. Their early years of gene therapy, people did some sort of, in retrospect, even at the time, they were perceived as quite crazy experiments. And I suspect probably people have tried, but are not using the state-of-the-art techniques to get any effectiveness. But that's just my supposition. But that doesn't mean that in the near future that somebody might, or an organization or a non-state sponsored group or even a state sponsored group might put together a team to do this. And I think what WADA is rightly doing is thinking ahead about how one would monitor and detect that if it did happen.
So when you say that you think there's other ways of gene doping aside from gene editing, what do you mean by that?
So there's two major ways of doing gene therapy right now. One is to take cells out of the body, your own cells out of the body, which can really only be done with blood cells, although it can be done with skin cells to some extent, and use what we call a lentiviral vector, which is a virus in which you've removed the viral genes and you've put in your gene of interest. And the lentiviral vector gets into the cell and integrates that gene into the genome somewhere. We don't know where, but it goes in. So now the cell has a new gene. So one could imagine you could take blood cells out, introduce a new gene, then give the patient's own cells back, and now they would have cells that might be making a new protein. And people have done that in gene therapy for people who have genetic diseases, and it works. And it's actually one of the ways that, if you've heard of CAR T therapy, it's a way of giving T cells a better ability to kill cancer. Mm-hmm. So that's been working in diseases. And so people are thinking about, well, what if you substituted a gene that was designed to help a genetic disease or a disease, and instead put in a gene that might help performance. The other major approach to gene therapy is using a different type of virus called AAV. And AAV is a virus that many of us have been infected by, and we never knew it. So it's not considered a pathogenic virus in humans. Um, and again, you take out the normal genes in AAV and you replace them with the genes you want, and then you make a large quantity and you can put the virus into the bloodstream, and the virus will then circulate and get into cells, and the new gene will be in the cell. Now, it won't go into the genome; it'll stay outside the DNA, but it can still make new protein. Mm-hmm. And again, AAV has now been approved to treat a serious disease of the eye, and there are approvals on the way for different diseases like clotting disorders, and it's been approved for a genetic disease of brain development called spinal muscular atrophy. So it would be a way of, in the body, delivering a new gene to many cells throughout the body. So if you thought you had a gene that might increase your performance by having more of it, this would be a way to deliver it to the body.
Right. And so as far as we know so far, there's 3 possible methods. So the first 2 that you mentioned have already been done in gene therapy. The other one is genome editing, which could be the future.
Could be.
Of gene doping. So we just talked about introducing genes into the body that might be beneficial for performance. From your knowledge, what are some specific genes in the human DNA that might be desirable for gene dopers to introduce into their bodies, and what do they do?
Yeah, I think I don't want to speculate too much because, of course, we have to be careful. People are smarter than me, but I don't want to give anyone any ideas. But certainly some obvious ones that are out there is, we know that long distance, endurance athletes have used erythropoietin to increase their red blood cell mass so that their endurance is better. Mm-hmm. And obviously the Tour de France ended this weekend, and it was an incredibly exciting event. And, bicyclists and cross-country skiers and long-distance runners all can benefit by having a higher red blood cell mass because you can carry more oxygen. So, we know that dopers have used extra erythropoietin in different forms to try to create their extra red blood cell mass. Mm-hmm. And we know that the detection methods, though, are very good at being able to distinguish between whether you're just using your own erythropoietin that we all make. It's a natural hormone that our kidneys make, versus somebody who's giving themselves extra. So you could imagine a gene doper thinking about, well, instead of giving myself the hormone at regular intervals, I'll give myself a gene and just have it continuously produce erythropoietin. So that's one idea. So many of the same proteins that dopers are using periodically, like erythropoietin, one could think about, instead of doing it periodically, giving it once and having it there all the time. Of course, if a doper used an erythropoietin gene, it could be detected in the same way that we detect the periodic erythropoietin. So it might be very hard to see that, to figure out. You'd have to, a gene doper would have to figure out how to make it so it looked like it was natural erythropoietin, but that's very hard to do, because if you express natural erythropoietin, if you express extra erythropoietin, you'll suppress the natural erythropoietin, and so you may not get anywhere.
Right, and so that's one is EPO or EPO that could be desirable. So there's a few more out there, and they're out in books and stuff like that. And so if an idiot like me knows them, then I'm sure an athlete knows them too.
Exactly.
But so for example, like actin, the gene actin, myosin and ACE. Like, what do you think about those and their potential?
Yeah. I mean, I don't think actin can be there. By the way, we also realize that EPO and human growth hormone at too high levels are dangerous. Mm-hmm. And so for a lot of sorry, I have a little puppy.
Oh, that's
so for a lot of these, the gene doper would have to worry about making enough to be useful, but not making too much that they would end up causing short-term harm. Now, we know that one of the real challenges is that athletes are willing to compromise their long-term health for short-term benefit. So I think the sorry, let me just deal with it. Hey. I think she's barking at the leaf blower. Is in normal medical practice, most patients would not compromise too much on short-term benefit if they knew that was going to shorten their lifespan in any certain significant way. But it's clear from surveys of athletes that their calculus is very different. Mm-hmm. They state that they'll compromise that. So I think, but the fact is some of these genes that you might consider actually have short-term harm as well. So getting the dose right is going to be if you were going to do gene doping, it's not obvious how you could assure that you would get the right dose.
So one of the most essential, things to consider, we talk about it in sports science all the time, is like the interaction between genetics and the environment. We're talking about nature and nurture. And simply having a gene doesn't mean that it's, that gene is going to be expressed and it's going to take its effect if the right environment is not there. Do you mind just explaining a little bit about the interaction between the gene and the environment and how that might come into play for gene dopers?
Yeah, exactly. I mean, you could have, personally, this is my personal view. I'm not speaking on behalf of WADA or anyone else when I say this. And everything I'm saying here follows that caveat. I'm just speaking about my own personal beliefs. I think to be, say, an Olympic level athlete, you have in your genome a variety of variants that in and of themselves, they don't make you a great athlete, but in combination give you a higher probability of being able to perform better in whatever sport you are, whether it's endurance racing, whether it's strength, whether it's coordination. But that genetic makeup might give you, a probability that with the right training and environment, you can realize the potential. So the way I see it is that we all are born with a genetic potential. And actually, I don't believe that I do believe that we all have our variants that would make us great in whatever the genetic variants that might make us a great athlete might be just the wrong variants to make us a great musician or a great thinker or a great communicator. Mm-hmm. That they may be contradictory. But that just gives you a chance of having that it's still the environment and your ability to train correctly, to eat correctly, in this case of athletics, that's going to allow you to realize that potential. So it's only a potential.
Right. Because, sorry, go ahead.
Yeah. Well, and of course the example is no matter how much I would want to be an NBA basketball player, I'm 5'10". It's just not going to happen.
Right.
So, it gives you the potential, but you still have to put in all the hard work, and all the things you need to do to make your environment great to realize that potential. Sorry, you're going to follow up with a question.
Yeah. So the reason I was asking that question is because I've read about a few studies in mice, for example, where they introduce genes and they cause that, genome editing, but then the gene itself doesn't like activate, or doesn't express itself, or doesn't come into fruition because the environment, the mice did not grow up with that gene, so they haven't had time to interact with the environment and express it. So is that something that athletes also have to consider and worry about when they're considering doping?
Well, that's exactly right. It could be that the effect of a gene on your potential for athletic performance comes during a certain phase in your development, a window, let's call it, and that after that window, its effect is not there. So even though you see a variant of a gene that's enriched in gold medal winners in a certain sport, it may not be that they actually need that variant then, they needed it when they were 12. So absolutely, the developmental biology of performance and the role of genes and environment could absolutely be there.
Mm-hmm.
Yep.
And I guess it's kind of like just any other form of doping. Like even if you, let's say you take anabolic steroids,
Yeah.
sure, they're not genes that are expressing, but without the right training and eating and everything, it's just not gonna.
Well, and then the other thing, sorry, I realized I forgot to say one other thing to your first question is that the human body has incredible what we call homeostasis. That is, it keeps things in balance. So, like I talked about with erythropoietin and EPO, if I gave myself extra EPO, it would suppress my endogenous EPO because my body would sense I have more than enough red blood cells. And so that is also part of the reason that sometimes these mouse studies sort of show there was no effect is the mouse got the gene, but it compensated by downregulating another pathway, and so you ended up right where you were. The other issue that you're raising is sometimes you put a gene into a cell and the cell turns it off because it's like, this isn't good. I don't need this. And so it turns it off. And that's also going to be a very real, so you could go through the entire process and the cell just says, no, this isn't natural. I'm shutting you down. And cells do that a lot.
So there's a lot of barriers, it seems like, for an athlete who's considering to cheat using gene doping. Like there's a lot of obstacles in their way for them to successfully do it.
I think that's correct.
Yeah.
Yeah. I think that's right.
Is one of them the viral injections? Like, because as you mentioned earlier, you can introduce the genes into the cell by using a virus. Could that in itself be a dangerous process?
Absolutely. So we know that several patients who've gotten the virus have died from the immune reaction to the virus. And we also know that you have to give, just like every drug, let's call the virus a drug. If you go give too little, it doesn't have an effect. And to make enough of this virus to give to somebody actually is not an easy thing. You can't do it in your garage. It takes really specialized expertise to potentially make enough virus to even have an effect. And at the doses that have an effect, you can cause toxicity. So, for example, I mentioned the virus AAV, which can be used to deliver genes in the body, and it works quite well. Well, it turns out that about 40% of adults around, a little more, sometimes less, other times, depending on who's measuring, already have pre-existing immunity to the AAV. So if I had an antibody and I had T cells which help fight in viruses, and you gave me that AAV to deliver a gene doping gene, I might have an overwhelming inflammatory response to that virus and get either, if I was lucky, I would just get super sick. And if I was unlucky, I could die from it because I got so sick. So you're right. It's not just as simple as putting a gene in a virus and giving it to somebody and, et voila, it works. There's so many barriers to thinking about this.
Yeah, and the process is a lot more complicated too compared to, let's say, HGH, where I'm sure you can get some, crooked doctor to give you some injections. You can, easily do it. Not in this case. Yeah.
Yeah. No, you cannot go down to your local doctor. Now, the other thing like HGH and EPO, just to take two well-known examples, were of course approved drugs for serious medical conditions. And what they've been done is they've been repurposed for athletic dopers. They weren't developed for athletes to use. They were developed for patients to use, and then they've been repurposed. And that's why you can go to your doctor. Although HGH, you have to have a special license to prescribe. I don't have a special, even though an MD, I can't prescribe HGH. I don't have that special license. Now, doctors can get around that and so on and so forth. So, one of the things people, when I look at the gene therapies that are developing for patients, none of them are obviously, none of them would help an athlete. They wouldn't be like, oh, if I just got that, that would make me perform better. So right now, I'm not aware of any gene therapy being developed for a disease that actually could be easily repurposed for athletes.
Right. And the ones that I've seen and come across are ones, again, like that help with medical disorders and stuff like that, that could potentially be taken advantage of. Some of them that increase muscle mass for people with muscular dystrophy.
Dystrophy. Yep.
You could use that, or for blood pressure. People have blood pressure. You want to dilate the vessels to deliver more oxygen. They use that.
Okay, so that must be where the ACE is coming from. Yeah,
right.
That would be a very subtle effect, I have to admit, because again, homeostasis is a very powerful. Our kidneys and our brains and our tissues sense how much blood is flowing to them, and you could try to dilate them, but the body may just say, nope, I don't need that much blood. I'm going to dial it back. So, now the issue of muscular dystrophy is the one that's closest. Is people are working on therapies that prevent the degeneration of muscles that occurs in muscular dystrophy. And people have speculated that same therapy could be repurposed in somebody with normal muscles to make them bigger. Now, the other gene that's out there is and I'm sure you have heard about it, is this gene called myostatin, where if you mutate myostatin, your muscles get very big, and I'm sure you and your listeners have all gone on the internet and looked at myostatin defective cows and myostatin defective babies.
Yeah.
It's not clear to me that that's healthy at all. And because it's so uncontrolled, it's not clear to me that while it looks, the cows look pretty impressive, no evidence that they're actually going to be better than a cow who worked out well.
Right.
To use cows.
Yeah. So what does the myostatin do exactly? Like, what's its mechanism in the body that causes this?
Yeah, I'm not a muscle physiologist expert, but generally myostatin. So the way we can break it, myo means muscle, statin means inhibitory. So what it is a protein that inhibits muscle growth. And that actually allows our muscles to, I think, be appropriately sized for humans. But you remove the brake, so you remove the statin, and now there's no inhibition on muscle growth, and your muscles will grow beyond what's probably healthy.
Right. But you're right about, simply increasing muscle size doesn't mean that you can put that to use in an effective way. You know what I mean?
Right. Exactly. Yeah. Exactly.
So if we go back just a little bit to catching the people who might use gene doping. So you mentioned earlier, if they use gene doping to increase erythropoietin, then you could use the same method that you already use to catch EPO doping for that. So is that going to be the primary method of catching athletes who gene dope? Is it just like to look for the same biomarkers or something? Or is there going to be like another detection method that you have to come up with?
I think, again, I don't want to go into too many details here. But I think the way WADA is thinking about this is, of course, you want to develop redundant mechanisms to try to detect people. And what we know is that in patients who've gone through gene therapy, they leave a signature that they've gone through gene therapy that can be detected. Now, usually when somebody goes through gene therapy, it's not a secret. So we don't care much about that signature. But I think we are learning from those patients what the signature looks like. And now if somebody didn't want to get caught, the question is can we from their blood and it's probably going to have to be from their blood, see that same signature. So I would say hopefully that's a specific enough, but still vague enough idea how WADA and those of us advising them are thinking about how you might see whether somebody has undergone gene doping. The gene itself, we've talked about some candidates out there, but one could imagine you can't think of all the different candidate genes that one might use. And so certainly gene-specific techniques probably should be developed, but you also want to have techniques that don't care about the gene itself, but they care about detecting the process.
Right. All right. I won't follow up on the signatures but maybe I'll wait like 10 years until it's already
out there. You'll start hearing about them.
Yeah, and then I'll start hearing about it. But know that I was watching Icarus the other day. I don't know if you've seen it on Netflix.
Yeah, I haven't seen it. No.
Oh, you should. It's about WADA and catching the Russian team that was doping.
Oh yeah.
Yeah, and they were talking about all the detection methods, stuff like that. So that got me wondering. So when it comes to PEDs, there's a long list of banned substances, obviously, but there's also a list of prohibited substances, like special cases, for example, like testosterone replacement therapy. WADA will allow it if you need it. HGH will allow it, if you need it. Yeah. Yep. Do you foresee in the future, when gene editing is actually efficient and it's working well, that maybe WADA will be like, hey, this is a list of genes that you're allowed to, as long as it's monitored and it's for medical purposes and all that? Or do you think that's too far of a...
Well, so my feeling, and this goes a little bit about to the issue of embryo editing, is that I sort of like the concept that you might consider using gene edit. If, let's say, it becomes safe and effective, you might considering developing editing up to what's normal, but that creating changes that go beyond quote normal would not be supported. And by normal, I define normal as the variants that are common in the human population. And we can discuss what common means, but I think it's something that we all sort of have a gut instinct. If 5 people in the world have it, that's not common. If the question is if say 30% of people in China have it, is that common or not? Well, we can discuss whether that's common or not. It might be, might not be. The other thing that I think we really have to be careful about is we started out talking a little bit about gene-environment interaction. There's also gene-gene interactions, which that genes don't develop variants in a vacuum. They develop them in the context of the other variants that are going on. And so even if you said, oh, this variant looks like it might confer some benefit, what you don't know is, does it only confer benefit in one circumstance but not in another? And that circumstance could be the other genes or it could be another environment. And so I think, again, another cautionary note that looking at a gene or a variant of a gene in isolation, it could be very misleading.
Right. And so introducing one gene could suppress another, for example, or cause it to mutate in a harmful way?
Well, could be, but I'm more thinking about, let's say you have a gene that, again, let's just use EPO as an example. You say, okay, there's a variant in the EPO receptor that makes it more active. And there are variants out there. We know that. We know those. What if that you only can carry that active EPO variant if you have another variant that downregulates the system, that homeostasis idea? So now if I don't have that variant that downregulates the system, and now I put in the EPO receptor variant that upregulates the system, all of a sudden I end up with a hematocrit of 60 and I have a heart attack because I don't have the right brakes that go along with the right accelerator.
Right. Right.
Does that make sense?
Yeah. No, that makes a lot of sense. Again, like you said, the genes aren't operating in a vacuum. They're operating in the context of all the other genes surrounding them that are interacting with each other.
Exactly.
Right.
Exactly.
So, given all those barriers and obstacles and like potential dangers and the gene doping, do you, in your opinion, not necessarily as a WADA consultant or anything, but in your opinion, do you foresee genome editing, or let's say, do you foresee gene doping as actually being the future of doping, or is that something like we glorify in movies and books and we like to think about it as like this cool thing?
I think it will happen. I think there will be. I think there will happen. As much as I've described all the barriers and challenges and risks and to it, the fact that I think it will happen, and I even think that it will. It may even turn out that in a small number of people they get some benefit from it. Mm-hmm. But I also think that it won't. Maybe I'm just being an optimist here, and I don't appreciate, But I also think it probably won't be widespread because of all those barriers and all the challenges. Whereas I think, in all honesty, while it's inconvenient to give yourself small doses of a performance-enhancing drug over and over again, it's actually safer and more effective and allows you to tailor your therapy better. Whereas gene doping is really a one-side. You don't get to really control the dose. You don't know what you're going to get out of it. I think it's going to be very hard to dial in just the right amount. Whereas when you're using these more, quote, standard forms, you can dial in the right amount for you at the right time, to be honest. Right. So, yeah.
Yeah. It just seems like when you were saying that you think in the future, yes, it might happen, it will happen. And that just goes back to, I feel like every medical advancement that happens, as far as like therapy and curing diseases, there's someone on the side watching and like copying what's going on so that they can apply it to doping. Yeah. And I guess if medical advances keep happening in gene editing, gene therapy, then that could be.
And that's why I think you're right. But that's why I think, WADA and others are doing the right thing by thinking ahead and saying, okay, we shouldn't rely on the fact that it's going to be challenging to say that it won't happen, but instead think about, all right, how do we put in redundant detection mechanisms so that if we miss with one, we pick it up with another.
Right. So let's end on a little bit of a positive note after talking about cheating and doping and want to just ask you a couple questions about your medical practice and your work with outside of WADA. So in your medical practice, and you and your lab at Stanford, you guys work on gene therapy, obviously, to treat diseases. So do you mind explaining like what kind of diseases you guys are working on and what kind of results you're seeing?
Sure. I know. I'm excited about that. So like I said, I got involved with the field because of meeting patients and taking care of patients who were born through, no fault of their own, specific variants that cause a disease. So I like to be really careful about, I think there are variants that, we all carry variants. If your genome and my genome are mostly the same, but we all have subtle variants. Even my genome in one cell is very different than my genome in another cell. And you develop new variants. But there are certain variants that don't just give probability of disease or performance. There's certain variants that are so detrimental it leads inevitably to a disease. And so one of the most common variants that people know of is a mutation in the beta globin gene that causes sickle cell disease. So sickle cell disease is a genetic disease of the blood. It's autosomal recessive, which means you have to have both copies have to have the mutation. And when you do, your red blood cells, instead of being able to effectively deliver oxygen to all the tissues in the body, can become stiff and they don't get through the blood vessels. It's exactly the opposite of what a gene doper, an athletic doper would want to do. It is by no way do they want to do this. So these patients get these blockages, and so they end up with all their organs in their body being damaged because the red blood cells aren't functioning correctly. So what we've been working on for many years is, can we take the patient's own blood stem cells? So the blood stem cells give rise to the red blood cells. Red blood cells, as you know, don't live forever. They only live, for a couple months, few months. And then they have to be replenished from the stem cell. So our idea is let's take out the patient's own stem cells, fix the mutation that causes the disease, and then give them back. And we've been working on that many years, and we hope that we'll treat our first patient using that in the beginning part of 2021.
So that's soon.
That's soon, yeah. Wow. We're putting together all our paperwork, and of course, before you start a trial like that, it has to be reviewed in the U.S. by the FDA to make sure that the risk benefit is appropriate for the patients you might enroll. So we think so. It's been a career-long endeavor, but of course, if it works for sickle cell disease, let me rephrase that. When it works for sickle cell disease, because I have to be an optimist about it.
Yeah.
There are hundreds, if not thousands, of other genetic diseases that one might be able to fix the same way. This patient has this defect that causes their immune system not to work. This patient has this defect that causes them to bleed. This patient has this that causes them to be prone to infections. And now we can go in and do the same process. It's like, okay, we know your mutation. Let's fix it in your own cells, give your own cells back, and now you'll be able to fight infections or be able to make sure you don't bleed or excessively. So I think optimistically, and again, been working on this for a while. I think we're really at the start. There's been successes in gene therapy, and there'll be continue to be more successes in gene editing. We're going to start seeing even more successes, and this idea that cells, our own cells, are the drug, is something that's going to happen, I think, even more and more over the next 5, 10, 15, 20 years. So we won't think about a small molecule or a protein as a drug, but we'll think about the cell as a drug. So I think, going back to the gene doping idea, so now we're going to think about, well, can people use cells as drugs? And of course, we have to think about how one might to enhance athletic performance. So, yeah.
Right.
But it's very exciting to be at this cusp where we can, really take people who have diseases that shorten their lifespan, cause frequent, pain and suffering, and think about using gene editing to fix a mutation and maybe even cure them. So we'll see.
No, because that topic is so interesting, and I have someone very close to me with rheumatoid arthritis, and so obviously the causes of what caused the disease to happen is unknown so far, but her rheumatologist was telling her, like, she thinks it's that so she experienced like a death in the family, and she thinks that is actually what it caused the gene to express itself, causing rheumatoid arthritis to happen. And so to think about all the potential of gene therapy to cure all the different diseases is actually really, it's a very positive thing to think about.
Yeah, and I think rheumatoid arthritis is an interesting example in the sense that probably there's a few people who have mutations where it's inevitable they get an autoimmune diseases, but most of us live in a situation where we have a probability of getting an autoimmune disease or not. And then a trigger or some environmental event pushes us over the edge. And so as genome editing gets better and safer, what we might do then is say, okay, you have a high probability of getting an autoimmune disease. Maybe what we can do is reduce your probability by changing some by specifically changing a variant or a variant in a specific gene.
Right. Yeah.
But of course, the same variants that might predispose you to an autoimmune disease might be the same variants that allow you to fight SARS-CoV-2 and COVID better. So we always have to be weighing. Maybe in one situation it's bad, but in another situation it's good. And until we know that, it's often hard to. I believe in being humble.
Yeah. So what was that this disease? I think that if you have it, you're immune to malaria, or maybe that gene that.
So sickle cell disease. Yeah. So that's it. So if you have one copy of the sickle cell gene and one non-sickle cell gene, you still get malaria, but you're less likely to die from malaria than if you don't have it. So the sickle cell gene we call S, and the non-sickle cell gene we call A. So if you're AA and I'm AA, if I got malaria, I would have a certain probability of dying from malaria. Actually, as an adult, I've never gotten malaria before. As an adult, I would have a very high probability if I got malaria at this age. But if you're AS, 1A, 1S gene, my probability of dying from malaria goes way down. Now, the problem is if you're SS, you have sickle cell disease, and you're even more susceptible to malaria. So it's kind of this weird shape, not weird, but you know what I mean?
Yeah.
The intermediate state is good, but the two ends aren't as good with respect to malaria.
Right. Yeah.
Yeah.
No, it's just
malaria is such a common parasite all over the world. The human population carries this S allele and some other variants to protect against malaria.
So one last question before we wrap up. I just want to get your thoughts on, there's a lot of companies out there that, they ask you to send a, sample of saliva or something, that's their DNA, and obviously you pay quite a bit of money for it, and they give you, a list of what you're susceptible to and whatnot. In your expertise, are those, for the most part, are those legit? Are those trustworthy, or do you think they're kind of taking advantage of people?
I think most of the companies are legit in the sense that they're giving you the results that are true. The interpretation of those results are much more difficult and subtle than perhaps the companies let on.
Right.
That, as I said, what they're reporting on is variants that increase your probability or not of having certain diseases. But humans are not very good with that probabilistic thinking about their health. We tend to be black and white, and what they're reporting on are not black and white. They're very subtle. You're predisposed or not. The other thing, though, I think is that they're also sitting on a gold mine now, because we've paid them, and now they have a bank of what the human genetic variation is. And we paid them for their bank. So it's a good business model.
Yeah,
but So do I trust that the results they tell you are true? Yeah, I trust the sequencing, the DNA results are true. Do I fully trust how they're going to use that data? I'm not sure.
Right. Yeah. I mean, if you thought Facebook has your data, wait until you see what those guys have in their.
Oh, I know, exactly.
Yeah, that's.
And wait until Facebook or them merge, and then.
Yeah, that's the next thing.
Yeah,
that's the next step. Awesome. Well, again, thanks so much for your time, and I really appreciate it. Just wonder if there's any, can people follow you on Twitter, follow the lab, follow your work somewhere? Is there a website they can track?
I'm not on Twitter, Facebook, or any of those. So, I'm not easy to follow, but, hopefully as our programs to treat patients move along, people will learn about it. But I don't have a natural place to follow. Sorry about that.
No, that's a smart idea. Stay away from all that nonsense.
Thank you. Well, thank you for inviting me to your podcast. This has been a very fun conversation. I appreciate it.
No, thank you. Yeah, I appreciate it. Thanks.